Okay, I think it's about time that we're ready to start this week's session. So I appreciate all of you who have joined me today. And I'm just going to share a screen with you so we can work through materials together. And you should be able to see the PowerPoint presentation that's labeled as the exam 1 review session. So that gives you an idea of what we will be doing today. But before we get started, I wanted to share with you that last week, quite a few of you got a chance to see that we had a little bit of technical issues, or not so little, with couple of things that stopped us from progressing ahead. So for that reason, we had to start late, and I have made the recording available already right after the session on Blackboard. So there was a chance to still catch up even if you had to move on to another appointment. What we're going to be doing today, I want to spend a little bit of time talking about the exam that we have scheduled for this week. And then on the Wednesday session, we'll get a chance to look through the material that we have for the weekly chapter material and for the lab this week. But what the exam will cover will be the first 2 chapters of our course, so the study of life and then the chemistry aspect that we had a look of. And if you recall, really the first chapter focused on discussing what biology is, what biology studies, and looking some of the general concepts such as what is the scientific method, how does the scientific method work, and so on. The second chapter focused really on the chemistry that forms the really foundational level for study of biology. So on this course, we will start from the smallest level and work increasingly upwards, having look of various level of studying biology and key concepts that set you up with the basic foundation that as you move ahead on your further studies. This week's exam, or this exam 1, is going to be due tomorrow, so on Tuesday, and you have all the way till the end of the working day, so there's no rush with getting it in as such. Many of you might wonder what sort of exams do we use on this course. So the exam is an open book exam, so you can use any of your notes, any of your textbook or other resources. So it is not going to be proctored. You do not need to use any online apps such as Respondus or a few other options that are available there. So you can approach it very relaxed. If you've taken good notes, you definitely are encouraged to rely on those. If you worked through the homework questions, the quizzes that we have for each chapter, you should be well set for success. So really my single one biggest advice for tackling the exam is that make sure that you have a look through the quizzes. If you missed any questions on those, go back to those and have a look of those so that you can refresh your memory of whatever might have been challenging. The test, however, does need to be completed as a single setting. So once you start the test, you should complete it on that one go. And since it is open book test, I've set a time limit, and the time limit is really not there to push you or rush you. It's going to be 2 hours that you have for it, and that's more than plenty of time. There's going to be 50 questions. They're mainly multiple choice, matching, filling the gap, that similar type of questions that you've seen before. So you have more than ample amount of time per each question. The time limit is there really so that you can't open it and then go do something else, come back, and then go and come back. So really the goal is that you sit down, get the test done, and then move on with other projects that you might have going on. If you end up with any challenges while sitting the test, for example, you get disconnected or so on, all you have to do is drop me a message and I can reset your test, so there's no worries in terms of that. So far, I haven't received any new accommodation plans, but if you have a documented disability, our college does offer student accessibility services, which helps you to come up with equal education opportunities, regardless of any disabilities or other challenges that might make the learning experience a little bit more challenging for you. So we level the playing field. It does need to be documented disability, and it all goes through the student accessibility services, not through me. So the details of the process have been shared with you on the syllabus. So if that applies to you, this is a really, really good time to get that taken care of. Cheryl Hernandez, who's the director of the Student Accessibility Services, is really good about getting these things turned around on a quick turnaround time. If you do think that you have sent me an accommodation plan and you haven't heard back from me, this is the good time to reach out to me again. I'm pretty much usually on top of those, but of course, I want to make sure that nothing slides past my radar. What I'm going to do now, I'm going to jump off from this presentation, and we're going to go to our Blackboard course. And there's something that I want to show you from there, and that's going to be our study guide. So I'm just going to jump to the similar view that you would have as a student on this class. And let's let the Blackboard load it up. So you should be seeing something very much like what we're seeing here. And when you scroll down the material, you'll see that right before the lecture exam 1, there is a study guide there. And this is what we'll use today to work through the material, to revise everything as it relates to the content that's being tested in the exam. So if you haven't had a chance to download that study guide yet, this is a good time to either open it or download it. What you'll notice is that the study guide contains gaps. And that's because usually if there is a gap, it's an important term or important concept. And I will go through those with you today on this session. And this session will be made available as a recording. And this is really the only time that I share the answers to that study guide. So I would really encourage you to either take your own notes, or if you're wanting to use that study guide, this is the good time to make sure that you have it available. What I'm going to do just momentarily, just for a few moments, I'm going to stop sharing the screen. I think that I've had few chat messages that have came through, so I just want to make sure that we get all of those looked through. So there's no worries whatsoever. I think that we had plenty of network issues last time. getting this class running. So you really haven't missed anything vital. We're just getting ready to work through the study guide. As you are getting your study guides opened on the screens, what I would ask you to do so that I can take the attendance is to post your name again on the chat. And if you have any questions, this is a really good time to just unmute yourself and let me know if you have a question, or you can write it on the chat. Really, this session is here to get you set up for the test. So I think I've written the study guide so that every single question that comes up in the test is covered on it. So there should be no curveballs. I really want to keep this course low stress and very, very relaxed. So there's no curveballs that I'm going to throw in the test. In fact, I think that the test questions follow very, very closely to what you've seen on the chapter questions. So if you have questions, let me know at any point. Since I'm not seeing any questions having came through in writing, or no one has shouted out any questions, what I'm gonna do, I'm just gonna pull up the study guide, and we're gonna use that to kind of remind ourselves of the material that we have covered so far and the material that might be coming up in the first exam. But like I said, at any point throughout this session, just give me a shout out if you have anything that you want to ask. So at this point, you should be able to see my screen, which shows the study guide. And on this particular version that I have pulled up here, I have actually filled in a lot of those gaps. So we're going to use that as we're working through the material that's important. Of course, there's sometimes a possibility that there's many correct answers to a question. So I'm just proposing some of those. It doesn't mean that it's the only possible way that you can answer to that question. So let's have a look of the first chapter, which was all about the study of life, and we found out that that was exactly what biology was all about. So we're looking at life, what makes something living, and we're looking at that in a scientific manner. So if you wanted to complete that heading that we have for chapter one way that you could complete it to make it a little bit more accurate would be to add there that it is all about the scientific study of life. So that brings us to the very first point that I had there when I asked that how could we define biology? And as I said, biology could be defined as scientific study of life. So we're looking at different forms of life, but also interactions of those life forms with other forms of life and with their environment. So the term environment that I have there really covers both the actual physical environment, but also all other living things in there. So what we ended up seeing very early on was that as such, since biology studied all types of things that relate to life. It ends up being a very large field that covers really many different sub areas. So I've listed some of them here. I'm sure that there's some that I may have overlooked, considering what might be the most relevant for you if you're heading to careers such as allied health or nursing or medicine or medical imaging. Those are some that my students often head to. Of course, anatomy, physiology would be very important, but also we want to pay attention on this course to things such as the chemical foundation of life. So basic chemistry is going to be important. We'll be looking at cell biology, which is known as cytology. We'll be looking at genetics as a form of or a field of biology that looks at tissues. That would be histology. And really, there's a wide range where we could head with this. The BIO182, which is the second series of this course that some of you might have been thinking of taking and might be needing to take it or might choose to take it, really takes us on a journey of evaluating different domains of life and different forms of life. And we're looking at a little bit of a larger scale than just cells and ethics. On that, we pay attention to things such as developmental biology in terms of the evolution. We'll talk about evolution, how that works. We'll look at various different populations or systems, how they all come together and work together. Many of you, if you are planning to go into healthcare fields, one of the preregs that many of you might end up finding is gonna be microbiology. So we offer a whole course on that. That's the Bio 205. And kind of to give you an idea what to expect on microbiology, we're looking at life in the smaller scale. So we're looking at various different kinds of pathogens that might be related to causing diseases or that might be beneficial to us. It is a fairly intense course, but it is a very, very useful course. So just something to put at the back of your mind of what to expect. So continuing on the chapter 1, we really started by noticing that if we look at the Earth and consider what's striking about Earth, it is the fact that a significant proportion of the Earth's surface area, and on this version I've said that it's about 70% of the Earth's surface area is actually formed by the seas. So it's not correct to say that the Earth really is a blue planet. So we're living on a blue planet. And the amount of the seas and the amount of water that covers the Earth's surface area is our first clue for having an idea of how important water really is for life. As a side note, just because I think it's always interesting to think of this, only about 5% of the seafloor of Earth has been mapped. Whereas if we look at, for example, the moon, all of the moon's surface has been mapped. And a lot of that work was actually done by the University of Arizona. They have been very, very successful on that work. And actually NASA turned to them for help when they were choosing the first first moon landing modules, landing areas. So the mapping that had been completed by U of A was very extensive and very, very useful. So I think it's interesting that we go to this space, we learn about other planets, but yet we know so very little about our own planet. The other thing that we end up noticing when we look at our own planet is that in addition to all the seas, we end up seeing a lot of rainforests that form a significant proportion of what we see when we look at our planet. And rainforests play significant role in our survival and the survival of the mankind on the earth. There's a twofold function that they do. First of all, we know that plants play a role in production of oxygen, and of course we as humans, we rely on getting oxygen into our body. That's what our cellular respiration requires. So our cells would not be able to function without that oxygen. So we're fully dependent on plants, especially providing that oxygen for us. The other thing that's going to be very important in terms of the plants, it is that plants play a role and major role in binding the carbon dioxide. And what we end up noticing is that the CO2 emissions are rising, and they have been rising for a long time. And it is fairly well shown that that is a direct consequence of the actions of the mankind. And it is really these rainforests that play a major role in binding the carbon dioxide. So if we end up in a situation where deforestation takes place, so we're losing all these big carbon dioxide binding sites on the earth, we are finding ourselves in a troubling situation where the carbon dioxide concentrations climb, and that will have then other consequences for the mankind's survival odds on the earth. CO2 emissions, for example, have been linked with the climate change and the overall increase on the global temperatures. But that's kind of to get us started and get to think about the planet we live on and how we fit in there. If we start looking at the development of the life, that is something that we discussed on our first chapter, we end up noticing that the very first forms of life on Earth were single-celled organisms that were found in water. So life evolved in water for the longest of time. We ended up having just single-celled organisms, so there was not really highly organized multicellular structures. So it's the single cell that served all the purposes for supporting life of that one cell and one cell only. First form of life has been mapped. We assume, based on our best knowledge, first forms of life appeared on the Earth about 3.8 billion years ago. In terms of the plants and humans, we have to go as close to the history, and this is really on bigger scale, fairly close, as 100 to 200 million years ago. And it is only about 2.5 million years ago when the first humans on the Earth appeared. So if you look at the whole history of our universe, of our Earth, and life on Earth, humans represent a very, very, very small part of that. What's something to consider about is, though, that the humans have had in that very short period of time that we've been on the Earth a significant impact on the entire planet. So we are having much greater impact on other life forms than any other life form before us has had. So that's something that comes with a responsibility and with things that we have to keep in mind. I always say in my classes that more you learn, more responsibilities you also are ending up with. So there's things that knowledge comes with responsibilities, what you do with that knowledge, and especially that you don't do bad and that you try to do good. One of the examples of simple forms of life that I shared with you was bacteria, and this bacteria that I talked with you about is known as E. coli. So Escherichia coli is a bacteria that's naturally found around in our, especially in our digestive tract, and it serves many important purposes there, such as playing a role in vitamin D absorption and utilizing the nutrients. So there's a lot of normal flora, normal bacteria that we should have present in the body and that we rely on. But we, of course, have heard, I think many of us think first thing when we hear about E. coli of the disease outbreaks that happen and that are associated with that. Well, there are virulent strains of E. coli that cause food poisonings. So there is good for it, but there's also negatives for it. And this brings me to the lab 0, or the first lab that we did, which was all about the lab safety. And actually in our lab, if we were face-to-face, we would be using E. coli as a model organism and to do various things that we've learned to do during this course, such as adding a gene into a cell or a cell's DNA, and that gene then changes the behavior of that cell. In this case, we would add something to E. coli, and what we would add is a gene that makes it glow in the dark or glow under the UV light. And of course, E. coli normally doesn't do that, but we will later on this course discuss how it is possible that we can do this kind of genetic engineering. And if we were now in a face-to-face lab, it would become very important that we practice safe lab practices so that no one ends up with diseases that could result, for example, from E. coli. The other thing that we talked on chapter 1 was that we had a look of the definitions of some of the most important terms that you end up seeing on this course. And of course, we talked about how biology was a scientific study of life. So we need to look at what is science then. And we saw that the term science came from Latin, and it was all to do with knowledge. And when we do science, we end up building up knowledge. So science and scientific process is all about using a systematic approach to build up our knowledge, come up with new knowledge, and further define the knowledge that we have. And of course, this could be then further divided in many ways. One ways that I showed were the general laws and principles of science that we base our work on. Well, science could be also further divided into very many other fields, and we are going to be focusing on this course on natural sciences. And natural sciences really look at the physical world. And natural sciences can be divided further into 2 groups, and those would be the physical sciences and life sciences. And of course, our topic of interest, biology, under the life science. Examples of physical sciences include, for example, the pure physics, chemistry, and so on. So those are very useful fields of science, but what we end up focusing on this course is all to do with a life science branch of natural sciences. Of course, we do have other fields of sciences such as social sciences. These are sometimes referred as softer sciences, if you wish. There's a whole different department that looks into these. And then there's some fields that fall between the physical and life sciences, for example. I think biochemistry is a good example of that. Another way of classifying science is to divide science based on the use of the knowledge that we gain. And we can talk about basic science, where we look at just the general facts about things, sometimes seen as pure science. This is the science that we do that doesn't necessarily translate into use immediately or directly. But when we look at fields of science that have immediate practical real-world uses, we're talking about applied sciences. So that's the other way of looking at sciences that you might recall from our first session. Then we talked about these scientific reasoning processes, and we saw that they were the scientific reasoning really aimed to draw conclusions, but do that in a way that's very systematic, very logical, that's following good practices. So we're really trying to be as objective as possible when we do scientific reasoning. And there were two approaches to this. In deductive reasoning, we took a general idea of something and applied it to a specific situation. So I used this triangle model earlier on when I discussed about this. And you can think of it as follows. So the very top of the triangle, if you wish, you can consider that as the really general laws or general understanding that we have about things. We know that those are what guides our thinking of the world. If you wish, you can consider them as laws or rules that we have accepted. And we can use these general laws, general ideas, and apply them to specific situations. So situations that we are at this very moment. And this would be an example of deductive reasoning. And because of the way how this works, sometimes it's also described as top-down logic. Well, there is an alternative approach, and the alternative approach is known as inductive reasoning. So in inductive reasoning, we're working our way towards broad general conclusions about life, about the world where we live in, but we come up with conclusions about the bigger conclusions about the world based on specific cases that we see. So if we look at this same diagram that I used as a model, in this case, we go from bottom up. So we're taking specific cases, which would represent this bottom level of this thinking process, and go to very general laws or general way of looking at the life. So that's the difference between deductive and inductive reasoning. And I did have a few different examples there that we looked during the class. So if you're still a little unclear of that, maybe jumping to those class videos to review the deductive and inductive reasoning might come in handy. The other kind of a theoretical concept that we had a look of was the difference between qualitative and quantitative reasoning. So there's many ways how we can do science, how we can approach the world, and qualitative sciences tend to be more descriptive. So we're looking at things, we're describing what we're seeing. Often they cannot be really these things that we're studying given numeric values. Qualitative way of classifying things. For example, we could talk about colors of cats. So we can have white cats, brown cats, black cats, and so on and so on. We can give a numeric value that, okay, if we code it as number 1 refers always to black cats, number 2 refers to white cats. That's totally made up a way of classifying things, because it doesn't mean that number 1 is better color than number 2, or that the difference between number 2 and number 4 would be the same as between number 1 and number 3. So it's just a way of describing things. It doesn't really have a numeric relevance to it. And the best way of really understanding what qualitative research is to have a look of what it is not. So when we talk about quantitative research, we talk about research that's all about numbers. So in quantitative research, we can actually come up with numeric outcomes of the topics that we're studying. So we can, for example, come up with results like most of the whatever we're studying are this kind or have this sort of a value, the amount of normal spread from a typical value is this much. We can categorize things. So it's all to do with playing with numbers. So we're quantifying what we're studying. And as a researcher, I've spent majority of my career. Go ahead. Was there questions? No, but as a researcher, I spent most of my career on looking at the numeric research. So I'm all about having some hard data. This is a very general way of kind of highlighting the differences between qualitative and quantitative research. There's a lot more to it, and I think that I prepared a rather nice table for us for this class that compared the qualitative and quantitative research. And I provided that table also to you on this study guide. In case there were a question in exam where you would need to be comparing and contrasting that. So you can kind of refer back to this resource to help you. So I'm not going to go through the greater details of the similarities and differences between qualitative and quantitative. That's something that's available for you there. But again, if you have any questions, I'll be more than happy to try to kind of fill in the gaps. And to kind of wrap up the first chapter that we had a look of, I think we should talk a little bit about the scientific method, because that is going to be something that's going to be really important for us to have a handle of understanding what scientific method includes and how the scientific method proceeds. So scientific method is a systematic method of approaching the process of doing science, so producing new knowledge. And it aims to be very objective, very carefully defined process, so we could eliminate a lot of errors that otherwise might come up in the process of doing science. So I've listed here the steps of the scientific method, and there might be a little bit of variation with the number of steps and types of steps depending on what source you go to, but in general, this is the kind of broad review of what you can expect to see when you review how scientific process works. So we have to start by either having some sort of a previous knowledge about the topic, or this previous knowledge might not be that we have a theory or we have a science about that topic. It might be simply that we've made observations that this typically happens, this typically doesn't happen. So it gives us some sort of a starting point for having an idea of where our interest to this topic comes from, what we might want to study, and what we might expect to see. So really, as the second step of the process, based on this previous knowledge, based on the observations, we want to come up with a question. And this question is really what we're studying. It's a research question. And I think we used an example of monkeys eating bananas. We had been in that theoretical example in the jungle, seeing that monkeys loved eating bananas. There's a few different ways of eating bananas, and we tended to see that monkeys preferred one way or another. So this triggered in our example that we used question of an interest, that is that way of eating bananas the ideal way? Is it the best way of eating bananas? Well, once we have our question of interest, we want to speculate. We want to come up with hypotheses of what is it that we're expecting to see. And you remember that hypotheses could have been seen as tentative explanation or a preliminary idea of what might happen. So if you want to put it in another way, you can consider it as an educated guess. It's what we're expecting to see. And you remember from our lab that I told you that a good hypothesis tends to follow the format of if something takes place, then something happens. So we're really trying to be very specific. And I'm sorry, my cat is just getting lonely and begging for a little bit of attention. So that's what I was reaching down to. So it gives us some sort of preliminary idea of what to expect. You can consider it almost as the foundation that then becomes a prediction of what we might expect to see. Well, these are all good steps to do and important steps to do, but really if we're doing science, we're coming up with knowledge, we do need to conduct some sort of an experiment. And this experiment can be in a lab setting, very formalized, very objective, very carefully defined, or it can be if we are doing more of a qualitative research. It can be, for example, going to the jungle, observing these monkeys, taking notes about them, interacting with them. So experiment doesn't have to mean that it is always in a lab setting. But in the scientific process, we do have that step where we're collecting data as part of the experiment. Oh, and I didn't mean to do that. So what data is, it can be often numeric information, but it doesn't mean that it is always going to be. Like I said, it could be also descriptions, it could be observations that cannot be coded in a numeric manner. But we are left as after conducting the experiment with some data that we want to analyze, and we are basing our further steps of this process on. So when we have our data, the next step after that is to summarize our findings. And this would be coming up with results. So based on the data, we can find that this and this is common, or this and this is not common, or these sort of things happen. So it really is simply a summary of that data, kind of compressing it, giving us, if we are using numeric data, what's typical, how typical it is, and by how typical I mean how much variation there is around that most typical value. But it can be also talking about categories that have no relationship into each other. But the big thing here is to notice that the results section of a scientific study does not include any explanations for these numbers that we're getting or these findings that we're getting. It's simply a summary of the data. And it is only within the section of the conclusions where we start to give explanations for what we saw. So if we are writing, for example, a good lab report or scientific publication, the results section actually ends up often being kind of a shorter one. It just tells us what we found out, gives us a summary of the data. And it is only when we get to the conclusions that we're starting to then expand on that and give explanations and connect it to the theory that we might have or other knowledge that might help why we're finding these kinds of results. And sometimes, of course, we don't have an explanation for our results. In that case, we do the best we can, but it might be the case that we don't have a definite answer to what we've seen. Anyways, at this point, once we have done our conclusions, one of the things that I think is really nice about the scientific process is that we go back to where we started. And how we go back is that we go back and look at our hypotheses. We want to see that do our results and conclusions support what we had as a hypothesis? Or do they reject what we had as a hypothesis? And this is the step that students often get really worried about, that your study, your findings should be supported, or they should be supporting hypothesis. This is the way we start. And there's nothing wrong with finding that our hypothesis was incorrect or that it wasn't supported by your findings. That's what we were there to do. We were there to find out an answer about that. So there's nothing wrong with finding out that that indeed was the case. So if your hypothesis is supported, you typically just move ahead and then publish your findings. If it is rejected, you can still publish your findings. But one thing that we often end up doing is that if it's rejected, we go back to the drawing board and we come back to our hypotheses, and we might redefine those hypotheses. We might reformulate them and repeat the experiment in light of this new information of how we're approaching it. So again, there is nothing wrong with finding out that our hypothesis was not supported. A big important step, especially in my former careers, if I think, is that we share our findings. That can be publishing them on a journal, peer-reviewed journal, that might be going to a conference, that might be telling to colleagues, writing a textbook, all sorts of different ways. But really, doing this experiment is not going to help us if we don't share what we found. And of course, once we have multiple studies, multiple experiments that all point to some sort of a direction, we end up forming this larger construction of knowledge, if you wish. And this is what we know as theory. So theory is really more than the outcome of just one single study or one single experiment. It's something that's based on a bigger body of knowledge. So, but that's ultimately what we're doing. We're building towards that in the scientific process. And I did talk a little bit about accidental findings. A lot of the time, if we use scientific method, we end up stumbling across with something that we were not expecting. And I think I shared with you an example of how Viagra was invented or discovered. Penicillin would be another good example. I've spoken about that on our chapter videos. We didn't get to do that in our face-to-face video. And final, really the big thing that I wanted to highlight on this one, on this chapter, was to talk about what makes something living. So we've talked about that biology is all about study of living things, but what is it to be alive? What makes something living? Well, I built a list of 9 points here. And again, there can be a little bit of variation on the number of points, depending on the source that you look at. But these are the things that we generally tend to find out when we're talking about what makes something living. So living things show some kind of an order. So there might be an order in terms of that cells are organized in an ordered manner. They form tissues, which then form organs, and these organs can form organ systems, and organ systems all together make up an organism, so the living thing, such as human. The order can be shown in many other ways as well, but typically living things are very much ordered. The other thing that we notice is that living things are capable of responding to their surroundings, to the environment where they're at. It doesn't have to be a human that we respond. Say you sit there and knock on your shoulder, and eventually you will get fed up with me knocking on your shoulder, and you will respond to that one way or another. It doesn't have to be animals that just respond. Think of plants, for example. Think of flowers. They open their petals for the daytime when the actual bees and other things are around, and they close for the nighttime when that's really not beneficial to have the flower open. Flowers tend to respond to environment. They tend to lean, or plants in general, lean towards the light source. Anyone who has had a plant on their windowsill knows that you kind of end up having to turn it around if you don't want it to just grow to one direction, facing to one direction. The other thing that's very typical for life is that living forms are able to reproduce. So we need to be able to come up with the next generation and next generation after that, and we come from somewhere. So reproduction doesn't have to be sexual reproduction, as we see in case of humans. So in sexual reproduction, we have a male and female counterpart, and the genetic material from both gets mixed, so we get a unique new individual. We can also have asexual reproduction. And in asexual reproduction, basically a good example of that would be a plant that grows a pod out of its side, and then that pod breaks off, and now we have two plants, but they are genetically identical of each other. So there's many reproduction strategies, and sometimes it can be a mixture of two, depending on conditions, whatever the organism goes for. But we do need to come from somewhere, and we try to continue our place in the life if we're a form of life. We try to pass the life onwards. Another thing that we end up noticing is that living forms or forms of life are able to adapt. They're able to adapt to their environment, able to conditions, they're able to adapt to a lot of things. That's really, adaptation is how evolution works. So evolution takes place over multiple generations. it doesn't just happen in response of something immediately. It takes generation after generation after generation to pass characteristics that makes an individual more successful than its peers. And once those characteristics build up, then that characteristic becomes more dominant because these individuals with these characteristics end up having more success, especially reproductive success, so passing their genes onwards. So you can think of at this stage adaptation as a tool of how evolution works. But the whole discussion about evolution, adaptation, all that, we go much more into that on our BIO182 if you end up taking that. Another thing that's characteristic for living forms is that we grow and we develop. So a lot of living things start small and they grow as part of their life cycle. But it's not just about growing in size, we also develop. If you think of a baby, newborn child, by the time that child goes to school, there's been a significant amount of not just growth, but also development of that individual. So that now it has gained skills, knowledge, and so on and so on. So it's really a much more than just physical growth that takes place. Well, the sixth thing that I have here is that living things are able to regulate themselves typically. So you are able to respond, oh, I'm not going to get overly heated. I'm not going to get too cold. We have ways of dealing with that, regulating, for example, in this example, our internal temperature, so that we keep it within a range that's suitable for our life. So we're able to regulate. We're able to regulate our responses. We're able to regulate our behaviors, our actions, and so on. So life bases on that. The other thing that I have here is that living forms get energy, process it, and use it to something. We get energy by eating things and utilizing those nutrients, that chemical energy that's stored in the food that we eat. We are able to make use of that chemical energy. We can store it or we can use it immediately. We can also use it later on if we need to do many different things, to do many different processes that we have just talked about. So really being able to use and process energy is an essential characteristic of life. And the last one that I have here is the concept of homeostasis. And homeostasis was all about maintaining consistent internal environment. So for example, humans, we have to have a certain body temperature. Getting too hot or getting too cold can mean death. So we do things to maintain that internal environment and keep it consistent for us. So that's what I mean by homeostasis. And that really concludes everything that I had from the first chapter. Do you have any questions about anything that we have covered so far? And I'm going to put this back on in a little bit. I just wanted to check. I see that there's a hand up. Erica, do you have any question? Can I help you with anything?
Yeah, on the scientific method, I missed number 12. Which did you write 12?
Let's go back to that. Absolutely. I so appreciate that you told. I might have gone too fast. I'm sorry about that. So let me just go back to that. Let's see. Hold on. I'm trying to share my screen. This seems better. So you should be able to see my screen. And if we go to the scientific method, it really, the number 12, the last step, was all about coming up with a theory. So we talked about how these many different experiments, the whole consensus outcome that we get from these experiments is the foundation what we build our theory on. So I hope that that helps. Ask me again if that doesn't make sense. I'll be more than happy to try to explain that a little more. And I saw that there was another hand, so feel free to just unmute yourself and shout out.
I have the same question.
Okay, I'm sorry. I clearly was rushing through that part. I'm sorry about that.
That's
I
okay.
really appreciate it. I can guarantee if you have a question, someone else in that class was already thinking of the same. So kudos to both of you for asking that question. If we don't have any further questions about chapter 1, I would like to go through the chapter 2 just quickly so that we have covered everything. But I do realize that some of you might have to jump out in next 15 minutes or so. If you do, the recording of this session will be made available immediately after this class. But I do think that we can do this in 15 minutes. So I'm not rushing us, and I'm not wanting you not to ask questions, but if we don't have any other questions, I'm just going to move on to the chapter 2, which is all about this chemistry. And when we talked about the chemistry, we started by defining it. And of course, we know at this point that this course is going to be all about science. You're taking a course in biology, which is a field of science. And when we're talking about chemistry, we're talking about very small stuff. So we're talking about stuff that's smaller than a cell, smaller than organelles that make up the cell. So we're really talking about atoms, molecules, and so on. So really, it's not like there's definitely a correct, this would be the only way to define what chemistry is. This is really just me trying to kind of lead you to what we'll be talking about. So whatever helps you to think about what chemistry is, that's a good way to go about it. This is not the only way. But really what we're going to be looking at, we're going to be looking at composition of atoms and molecules, structure of them, how they behave, what properties they have, what kind of reactions they can make. So there's many things that we can get out of this. And this really is the smallest level or the tiniest level of studying biology. We go to the smallest and then we start building on this course, the bigger and bigger. So what I wanted to do was to do some definitions to start with. And we started by defining what matter is. So matter, the classical or traditional way of defining it is by saying that anything that takes up space and has a mass is matter. So this pen, for example, will be matter because it takes up a space. We've got clear space that it takes in the universe, and it has a mass. And the term mass sometimes throws off some students because I think we're much more used to talking about weight than we are to talk about mass. So weight is the same thing as mass, but weight is dependent on the gravity. So there's a different gravity on Earth than there is on the moon. So if you say that, oh, I feel that I weigh a little bit too much, well, the one way to deal with weighing a little bit too much is just to move to the moon. You will weigh less there because moon has a lesser gravity, so your weight will be less. However, your mass would remain the same. So mass is just a little bit more scientific way of looking at the weight. It's not dependent on where we're measuring that weight. The other thing that that we saw, we talked about matter taking space. So anything that's matter has a volume. So volume is really the space that that thing takes in the universe. And we often end up measuring volume of things, for example, by submerging them into water and looking how much of the water they end up pushing up when they're submerged and so on. So we can end up measuring both the weight or the mass and also the volume of an object. So if it has both of those, it's going to be matter. And matter can take many different forms. Matter can be solid. It can be liquid, it can be gas, or there is a fourth one that we really don't get to talk too much about, but plasma is the fourth form of matter that it can take. And of course, plasma is not a form that's available for all things that we might study, but for many things. So as long as you're able to think of the solid, liquid, and gaseous forms that matter can take, you're doing pretty well. And I have there examples for each one if you're a little bit confused by meaning. In this one, I've used water as an example of matter and different forms that water can take. When we should also talk about elements. So elements are smallest form of matter, if you wish. So elements are when you cannot take or break down something into smaller by just normal chemical means that we have. These days we can go far beyond what we used to think is the limit. So that's why I'm highlighting the normal chemical means. We can do a lot more than we thought we can, but elements are really the simplest, the most basic forms of matter. And all life, all of our universe is made of various different elements. And there are 4 elements that are the most important ones for us to know. And these are going to be carbon, hydrogen, nitrogen, and oxygen. And actually 96% of you is made by these 4 elements. So the remaining 4% is other stuff, but you are made of mainly of carbon, hydrogen, nitrogen, and oxygen. And it really is the carbon and hydrogen that are going to be the most important when we study organic chemistry. So organic chemistry is based on study of carbon and hydrogen and then other stuff that's attached to those. So that's the definition of elements. And that brings us to talk about the periodic table of elements. And you remember the periodic table of elements gave us a list of all known elements that we have, that we know of. And in addition, periodic table of elements gave us a little bit more information than just the name of that element. It gave the atom number, electron configuration, also the chemical properties of that element can be read from this table. So it's going to be a really, really useful tool for us. And we ended up finding that there's 92 naturally occurring elements. And then in addition to that, we have actually 26 elements that can be made in a lab, but don't really exist naturally or don't remain around in most of the normal settings. So you need to do something funky to make those happen. So that gives us 118 elements that we're going to be interested in. And all of those 118 are listed on periodic table of elements. And you'll notice that there is kind of a sub part there that we've added stuff that wouldn't fit into our original periodic table of elements, as we found that it should fit in with that little gap that we had there. But we kept the shape of the periodic table of elements fairly similar to what we first saw. And we also remember that by using this periodic table of elements, we could tell whether something was very reactive or non-reactive. And that had to do all about the outermost electron shell of that element or of that atom. So that's why the periodic table of elements becomes very useful for us. And we saw that we called the rows in the periodic table of elements as groups and columns as periods. And we saw that on a periodic table of elements, typically what we have for each one of these, we have a little bit more information than just the name of the element. Typically, rather than writing the element all out, we use an atomic symbol for that element, and usually that is an abbreviation that comes from either Latin or English name for that element, but it can be sometimes a little bit harder to work out what it is. In addition, we typically end up seeing the atomic number and also atomic mass of that element for each of these elements on their boxes. So those would be useful things to know as for what we can find in the periodic table of elements for each of these elements. Then I wanted to talk a little bit about the atoms. So these are really the chemical elements, what we've been talking about, but a little bit of a different way of defining them. So this is really the smallest form that any normally existing matter can take. So the smallest unit of stuff that we find that the world is made of. And atoms are made of subatomic particles, so from 3 different kinds of things. And what atoms are made of is of electrons, which were located on the outermost shell of that, or were located around the atom's nucleus. We had multiple different kinds of shells. We could have one or more, and usually it was the outermost shell that we were interested on, but electrons are going to be on various different shells. So electrons circulate around around the atom's nucleus. So let's add an electron here on this diagram. And electrons, what's important to note about them is that they carry a negative charge. So they are negatively charged. Ooh, I'm losing my tools there, so let's pull them back up. And this, whether some subatomic particle has a negative or positive charge, it will become important in a little bit. So just keep that in mind, that electrons are always negatively charged. Well, the center of an atom is made of a nucleus. So just like a cell has a nucleus, so does an atom have a nucleus. And we have 2 different things that we find within the nucleus. We find protons, and we find also neutrons. And you might wonder, well, why does it matter? Why are we separating these 2 things? Why can't we just say that we find stuff there? Well, it comes back to this charge that different subatomic particles had. And let's have a look of the protons. They are positively charged, so they carry a charge that's an opposite to what the electron had. And then neutrons, instead, they had no charge whatsoever, so they're neutral, as the name would suggest. And typically, normally, we end up finding that the electrons and protons, their charges end up evening out each other. So electrons have a very small mass, whereas protons and neutrons have a larger mass. And this is why when we're calculating the mass of an atom, we really include only the mass of the proton and neutron. And we talked about the mass of the of an element earlier on here. And I do know that it's quarter past, so if you do need to go, I totally understand. I'm going to try to wrap up this section though. So if you can stay around, you stay around, but if you need to go, you go for it. When we had different number of neutrons for a same atom, but or same element, but they have the same number of protons, we have different isotopes of that particular element. And isotopes were especially important when we talked about the radioactive isotopes and radioactivity in general. And radioisotopes are the radioactive isotopes of an element. They are typically unstable combinations, so over time they end up breaking down, and they store energy within them. So this is where the radioactivity and the half-time and breakdown of radioactivity comes from. And I mentioned earlier on that typically you end up having the same number of electrons and protons. And if this is the case, this atom is now electrically neutral. So it's in balance between the pluses and minuses. So let me just add that there just as a reminder. So we had electrons had a negative charge and protons positive charge. So those would be in balance in an electrically neutral atom. Then we talked about the fact that different shells of an atom could contain different number of these electrons, and this follows a predetermined formula. So the first shell can contain maximum of 2 electrons, the second a maximum of 8, and the third one, it gets a little bit more complicated. For the purposes of our course, we're just going to think that it holds the maximum of 8. In fact, it turns out that the third shell can contain up to 18 electrons, but there's a little bit more that we need to know once we go to that path. So for purposes of our course, we're just going to stick with the three first electron shells, and we're going to consider that they contain 2, 8, and 8 electrons. And if you're curious about where this 18 comes from, I'm totally happy to provide you an explanation of that, but let's do it right after the class. So this is what you need to know. Stick to those 2, 8, and 8. The outermost shell of an atom was known as the valence shell. And this valence shell defined how an atom ended up behaving. If there was the full maximum number of electrons that could be fitted in there, this atom is going to be very stable. So it's not going to be looking to do a lot of chemical reactions. It just prefers to hang out on its own. It's got to the place where it wants to be, and it's happy being like that. We have some atoms that lack only a few or have only a few electrons on their outermost of their valence shell, and they want to try to either get rid of or fill that shell. So they're very reactive. So they end up doing a lot of chemical reactions. They're very eager to do that. So that's something to keep on mind. And that's the foundation for what we were looking. A couple of other terms. Molecule is formed when we have 2 or more atoms that bind together. And this binding of atoms is going to be important for us. So a molecule was when we have at least 2, we can have to a huge number of atoms that end up binding together. So they form one block together, one complete thing together. And then we talked about chemical reactions. So in chemical reactions, we end up rearranging the structure of the components that make up the whatever we're working on, the molecule or so on. So in chemical reactions, there's a lot of rearrangement taking place. And 2 terms that I wanted you to know was that whatever goes into that chemical reaction was known as a reactant, and whatever comes out of that chemical reaction was the product. So we can think of that we have something that we end up putting in, then the reaction takes place, and a new thing comes out of that process of reaction. So these things that we saw here going into the reaction were our reactants here, and what came out of the reaction was our product. So those are terms that I wanted you to know. And the reason why we talked about these bonds forming, things getting together, was that we had different kinds of bonds that can take place. And I want to go through these even though we're pushing it for the time. So covalent bonds were based on atoms sharing some electrons so that everyone that forms that molecule that's formed by these covalent bonds is happy. And they're happy by having that full valence shell. But to achieve that full valence shell, they need to share some of the electrons. amongst them. So none of them has enough quite on their own. And what I wanted to do was to show different kinds of covalent bonds that can happen. So here we have two atoms that I've drawn, and let me get my drawing tools back. And if these two atoms end up having to share an electron each, so there's one pair of electrons being shared, when we draw chemical formulas, we typically end up drawing that as a single line between these two different elements. If these atoms end up actually sharing two pairs of electrons, so in total 4 electrons is shared. Well, now in chemical shorthand, we symbolize that with a double line. And we can even end up having a triple line where in total 6 electrons, so 3 pairs of electrons are being shared. So just something that when you're reading those chemical shorthands, you can make sense of what's happening there. What we end up having is that sometimes these electrons are equally shared. And if they are equally shared, we call these as non-polar bonds. So they are equally shared. Both of these atoms get to hold that shared electron as long of a time. A lot of the time, though, it ends up being that we share these electrons unequally, and we end up with a polar molecule. Water is a good example of that, and we'll discuss about that in just a second. The second type of bonds that we discussed was that when we are not actually sharing stuff, but rather to achieve this full electron shell, we end up either giving away or receiving some electrons. So we're either losing or gaining electrons to get that full electron shell. And if that's the case, we talk about ionic bonds. And why this is important is that when an atom ends up giving away electron, it will lose one of those negative charges, but it will still have all of those positive charges on its nucleus that it had initially. So we end up with overall slightly positively charged atom. And now we talk about it being a cation. Oppositely, if an atom gains more electrons, now it's going to have more of these negative charges than it had positive charges. So overall, it's going to be slightly negatively charged, and we talk about anions. Well, what we learned in life, or learned in this class, nothing else, was that the positives attracted negatives, and negatives attracted positives. So opposites attract each other, and as a result of this overall positive and negative charge difference, these atoms end up packing next to each other. So this type of attachment between these atoms that's simply based on the slight difference between overall negative and overall positive charges is known as an ionic bond. And a lot of the salts are good examples of these. So most salts are made in this way. And finally, the last type of bond that I wanted to mention to you is hydrogen bond. And hydrogen bonds are really a special occasion or special case, and that's because water is so important for us that we have really taken this this own group. And what happened here is that we had a covalent bond between two hydrogens and one oxygen. And as they are sharing these electrons, we end up noticing that the electrons spend a little bit more time here at the oxygen's with the oxygen than they spend with the hydrogens. So overall, this molecule, water molecule, is slightly negatively charged at the oxygen end and slightly positively charged at the hydrogen end. So we are now seeing an example of a polar covalent bond in this case. Well, what happens is that when we have a bunch of these water molecules in a substance, well, this slight positive and negative charge difference causes all of these molecules to be attracted to each other, so that the slightly negative ends of the oxygens are attracted to another water molecule's slightly positive hydrogen end. So there's this connection between these water molecules that results simply from this unequal sharing of the electrons, which we saw in our first example of covalent bonds. So hydrogen bonds are just a special group because water is so important for us. And that brings us to just list the properties of water. Water is very essential for life. We end up finding that as far as we know, all living things are largely made of water, life evolved in water. We're about 70% water as humans, and we can't really think of that life would exist if there was no water. So that's why water is going to be really important for us. And 7 properties that relate to the water that I want you to know is that it is a great solvent. Most things dissolve in water. There is a high cohesion and adhesion. So we talked about the water molecules being attracted to each other, but also water molecules being attracted to other surfaces. That gave us a strong surface tension. So we remember, for example, bugs were able to walk on the surface of the water. Water stored heat extremely well, so we can store heat and release heat. That's why the climate is kind of more consistent on the coastal areas where we're by the water. In desert, we end up getting big temperature variations because there's not water. Water can take different forms, and we talked about those different forms earlier on. And water is excellent transport media. Think of the blood. Blood is largely water, so we're able to transport many things around the body in blood. Same goes for plants. Plants transport nutrients, but also many other things in the water that they pick up from their roots and transport throughout the plant. And finally, 2 things that relate to the water that we end up finding, because water is a polar molecule, that we end up getting certain molecules that are highly attracted to water. These molecules that are very attracted to water are known as hydrophilic. So they love water. And then we have other molecules that are hydrophobic. Well, if you think of what hydrophobic would mean, they try to avoid it. If you have a phobia of something, you definitely don't want to hang around with whatever you have a phobia of. You try to get away from it. If you have a phobia of spiders, you're not going to go and hang out with spiders. So those molecules that are hydrophobic, they avoid water. They do exactly that. They try to stay away from water. And other thing that we noticed, we talked about icebergs, that when water is liquid, the water molecules are more tightly attached to each other. But when we have ice, the molecules that form the water, or the water molecules, they end up taking a larger space between each other. So that's why the volume, or the same volume of water takes a larger space when it's frozen. That's why you shouldn't put a glass bottle that's closed into a freezer. It's going to expand and explode in a freezer. That's why, for example, a garden hose would break if it's filled with water and it doesn't get emptied and it gets freezing. Well, the water inside the garden hose will expand and it will break that garden hose. Then when the temperature drops, temperature rises again, the ice turns into water, but now we have a broken garden hose. I hope that that makes sense. And the very last thing that we discussed on the Chapter 2 was to do with the pH scale. And I'm expecting that you are familiar with the pH scale from the past. If you're not, there is a very good lecture video that I prepared on that chapter. So pH scale looked at the concentration of hydrogen ions in a solution. So it's simply a mathematical way for us to see how much hydrogen we have in a solution. And it ran from 0 to 14 with the neutral, so the middle point of the pH scale being 7. And we will be talking about buffers later on this course, so you will become much more familiar with them at that time. But now all you need to know is that what buffers do, they help us to maintain the pH within a certain range, within the narrow range. So we can add buffers to various solutions and they will bring the pH of that solution to whatever we want it to be. And that's how your body is able to maintain its pH. We have many buffers in our body. I do think that I also mentioned really briefly at the end of that chapter about the organic chemistry as being chemistry, where we are a field of chemistry where we're looking at the carbon and hydrogen atoms. So we're interested about carbon and hydrogen. That the carbon and hydrogen are really what all of the organic chemistry is based on. Then we have molecules that are made of carbon and hydrogen, and they have little things added to those. Those are side chains, but everything in organic chemistry is built around carbon and hydrogen. Isomers were different forms of the same molecules. They have the same chemical formula, same amount of same elements, but they are organized in different arrangements, so they can take many different shapes. That's again covered in the lecture video if you're kind of wondering. That doesn't sound familiar. And the functional groups are what I said, that most of the organic chemistry was made of carbon and hydrogen. They were the little groups that were added to these carbon and hydrogen to give chemical properties for these molecules. So that really completes everything I have for you on this study guide. At this point, I just want to make sure if you have any questions, if I've skipped anything, gone through anything too fast. So just feel free to either type it on the chat or unmute and shout out. I see hand. I can repeat what isomers were, and then Erica had a question. So let's go. Let me just share. So we go to isomers, and let me share the screen. So let's take an example of isomers. We have here molecule that's made of 3 carbons, and then we have hydrogens attached to these carbons. And I'm not going to draw every single hydrogen. You just know that there's hydrogens coming out of these carbons. So the big thing to notice here is that there's 4 carbon atoms. Well, we can take all the same combination of stuff that we saw here, and we can arrange it in another way. We still have 4 carbons, and then we can add our hydrogens to this molecule. And what we end up having is the same amount of carbons and same amount of hydrogens, but they are completely in a different shape in these 2 different molecules. These 2 different molecules that we're seeing here would be known as isomers of the same substance. So really what isomer means is that it's made of the same chemical components, but it's taken a different physical form. So I hope that that helps. I know that I had a really good slide about that, and I'm more than happy to pull it up if that's still you're kind of a little bit like, oh, that doesn't make sense. Just let me know. I'll pull up a slide to show a better example of that if you're still confused. But the big thing, compare here, 4 carbons and 4 carbons. On paper, if you just list the ingredients that made up these molecules, they look exactly the same. However, in reality, the shape that they have taken is very different. And their shape being very different will have consequence that these 2 molecules that we're seeing, they will behave very differently in a chemical sense when we're doing things with them, even though they're made of the exact same substance. So these would be isomers of the same molecule. So we have the same chemical ingredients, but they take different shape. I'm just going to jump back to see if that made any sense. So it is the same, it's made of same elements, but different form, different shape. So the reason why I'm not saying that they're the same element is because it's actually made of multiple elements. You remember each element was one of those letters. So it is made of same amount of exactly same elements, but they take different forms. What we can have is that an atom can change its shape around, and that molecule can change its shape around, and that molecule changing its shape around would be that molecule taking different forms of each other. So those would be different isomers. So your thinking is perfectly correct. Just a little thing with the words that we just don't say element. Oh, I'm glad if that made sense. I see that Brittany had her hand up, and I think someone else had earlier on. But Brittany, is there anything I can help you with?
Sorry, no, it was answered. Thank you.
Oh, I'm glad if that makes sense. Did anyone else had any questions at all? I know that we've stayed a little over time. Trust me, I'll give you extra credit for having stayed over time. Do you have any questions about anything at all that we've covered? Well, since you've stayed to the end, I'm going to let you into a secret. I always try to make it worthwhile to stick around. And let me just share a screen again. So when you are taking the exam this week, or this lecture exam one, I believe, and I'm not going to swear that this is definitely the case because I don't remember, but I believe that pretty much every single question that you will end up finding in that lecture exam was also asked in your quizzes. There might be 2 new ones, but at large, it's going to be the same stuff that you've already seen. I'm not throwing you any curveballs. So if you ace the quiz, you're good to go. If you didn't ace the quiz, go back to the quiz and look through. You can review your old quizzes, that where did you lose points? And the quizzes should also show you the correct answers. So if I was a student and I would know that this is coming up, I would go back and I would make sure that I know an answer to every single quiz question, because that's likely what the lecture exam is made of. I hope that that makes sense. Now, does anyone else have any questions? Oh, let me see. Will the study guide be a big help? Absolutely. Every single question that's in the exam is covered in that study guide. So if you have the study guide, I would hold it next to you when you're taking the exam. Every single question is answered in that. Perfect. If you have any questions, I'm going to be around here, so feel free to ask those. If you don't have any questions, you can leave, but if you could do me a favor and type in your name once more to the chat, so I know to give you extra credit when I grade the exam for the fact that you stuck around. So just drop your name to the chat and I will make sure that you get extra credit for your exam. And this is all I have for today. I know that we went overtime. I apologize for that. If you have questions, I will be sticking around. If you don't have any questions, this is it. And I hope you do well at the exam. I appreciate you having stuck around. And if you have any trouble with the exam, all you have to do is drop me a message. I'll be glued to my Blackboard for the rest of the next 2 days. So if you have any technical issues, I can just reset your test. Thank you. You guys have a good night. Thank you so much. I really do appreciate you having been around, and I hope you have a very good rest of the night. Of course, one more question. Will we cover chapter 3 and lab 3 this Thursday? Absolutely. So I will cover all of the chapter content for chapter 3. And let me just pull up. I just want to check what's in the lab. So do I cover it? I think I cover both on our Wednesday session. I'm just making sure that sometimes if the lab is very straightforward, I don't cover it in a greater detail. Yes, so the lab is going to be all about measuring and metric system. So it's going to be fairly straightforward. I will give a short review of the lab. We won't spend as much time as we would normally spend, but I will definitely cover it. I hope that that helps. Great. Thank you so much. You have a good night and you're doing great. Just hang in there. Yes, Erica.
Were you going to talk about, I forgot, at the end you were saying about the 18 electrons or like the 118 elements or something?
Of course, I can totally talk to you about that if you're curious. So really, when we go to look at does the third electron shell contain 8 or 18 electrons, that goes a little bit beyond to what we would normally cover in this course. So that's why I just didn't want to dump that as thing that you have to know. You don't have to know that. All you have to know is that we go with the rule of 2, 8, and 8. But why it is that sometimes we end up seeing that we might have more than those 8 on the third shell. So let me just pull up a really good way of explaining it in writing. So it comes down to that if you have no shells greater than 3, the third shell can hold maximum of 8. However, if you have fourth shell and so on and so on and so on, then the rules change a little bit. If you have a fourth shell, we can actually squeeze in up to 18 electrons on the third shell. So this is where it gets a little bit of kind of out of whack what would be logical or what would make sense based on the knowledge that we have covered so far, it actually does get a little bit, these rules get a little broken, but you don't have to worry it at this level. I don't know if that really answered it. I can give you a full explanation if you want, or are you happy with that?
I'm happy with that.
Okay, let's go with that. I'll prepare something in writing to provide the full explanation, but I would never expect you to know that. That would require you to have taken university level chemistry.
Okay. Okay, thanks.
Okay, good. Thank you. Have a good night. Good night. Thanks again. And that is going to complete our recording for this session. As always, I'm very easily reachable at the exam time, so don't hesitate to send me a message either on Blackboard, or I do try to monitor the email as well. But what's most important for me is that if you have a question, ask it. Don't feel that you have to take the exam without having gotten an answer to every single question that you possibly can have. And that concludes our review session for the exam 1. And I'll make this recording available for you in no time. Have a very good rest of the night. Bye.